So I want to introduce Dr. Michael Boyle. He's a research scientist, zoology, animal development, and molecular biology.
Wake up call.
That was one of the fish screaming. Anyway, so Dr. Boyle earned a PhD in zoology from the University of Hawaii at Manoa in 2010, where his research centered on the developmental biology of marine invertebrates. His specializations include embryology, molecular biology, and microscopy. He completed postdoctoral fellowships at the Smithsonian Marine Station in Florida and the Smithsonian Tropical Research Station, or Institute, in Panama. From 2015 to 2021, he was a staff biologist and principal investigator of the Life Histories program at the SMS, the Smithsonian Marine Station. Dr. Boyle then joined the Institute of Creation Research in 2022 as a research scientist, where now he oversees the biology, molecular biology, and microscopy laboratory. He has authored over 40 peer-reviewed publications, contributed to scientific books, and is lead editor of a symposium volume, ISBS2, an Atlas of Marine Invertebrate Larvae, 2nd edition, 2025. It's my pleasure to introduce Dr. Michael Boyle.
So, at the Institute for Creation Research, we have a mission statement. And that mission statement is to support the local church, which is you people right here today. And 3 points within that mission statement are applicable to what I'm going to talk about today. The first one is that we oppose the deification of nature by exposing Darwinian selectionism as an idolatrous worldview. In other words, natural selection according to evolution, nature becomes an idol. And all evolutionary biologists are actually worshiping that as an idol. Nature doesn't have a mind and it can't choose and select. So we see nature is not something that's valid in the world. The second one is that we're aiming to change Christians' view of biology by constructing an organism-focused theory of biological design that highlights Jesus' work as creator. So nature, no; Jesus, yes. Then we're also defending the gospel and countering objections to the Bible, or to the gospel, by equipping you with Bible-believing verses, science-affirming verses throughout the Bible. So that's how we equip you. And today I'm going to show you a lot of science that it's going to be strange to you. I'm going to try to make it as simple as possible. You're going to learn a whole bunch of things, and this is now the cutting-edge research, one of the most important programs in our research at ICR that's going on today. Why do we have this mission statement? Because we're at war. If you haven't noticed, we're at war for the souls of people. There's a war with Satan, there's a war with the lie, and here's the lie. Billions of years of nothing created everything by random variation, natural selection, and time. This is the broadest sweeping version, which includes evolutionary biology. First of all, there's nothing wrong with randomness. In fact, random is what's going on everywhere in the world. I don't think there's two of anything that are exactly the same. 2 electrons are not in the same place. No stars are the same. No people are the same. Random is just part of nature. But randomness can never produce order. It's always going towards disorder. Things are coming apart. Variation is a hallmark of God. Flowers, birds, tropical fish, people. No 2 things are the same, and the variation is what he loves. At the bottom of the ocean, there's all kinds of organisms with color that people don't see. He walks the depths of the sea, and he sees that variation. It's pleasing to him. Nature cannot choose, it cannot discriminate, it cannot distinguish, it has no mind, it can't think. Nature is just the sum of all the natural laws in the world. Gravity, motion, things like that. And time, at the ICR, we consider a straightforward reading of Genesis comes out to approximately 6,000 years. So that's the way we read Genesis. And we can get into that later on, but if you knock out this pillar of time, the entire evolutionary theory falls flat. There's no time to go from non-life to life, from non-living molecules to the first cell and all the way through to you and I today. You can't do that in 6,000 years. You can't do it in 6 million years. You couldn't do it in 4.6 billion years for a lot of reasons. And we can talk about that too later if you want. But we know the truth. For in 6 days the Lord made heaven and earth, the sea, and all that in them is, and rested the seventh day. All things were made by him, and without him nothing was made that has been made. For by him were all things created, that are in heaven and that are in earth, visible and invisible, whether they be thrones or dominions or principalities and powers. In fact, in all of these verses, and every other verse from Genesis to Revelation where our Creator talks about creation or being the Creator himself, he never gives creative ability to any other source, any other person, any other power. He's always the Creator all the way through. There's one Creator, and nature is not it. Jesus is our Creator. The evidence, continuous environmental tracking. This is a model we're testing right now within our theory of biological design. And what this says very simply is that every organism, every kind of organism at every level was created by God to actively and continuously assess the conditions in their environment and self-adjust to those conditions on every level. Biochemical, molecular, genetic, cells, tissues, organ systems, the entire organism is the one that's doing the change. Nature and the environment is not changing and adjusting and making new species. The organisms were designed to adjust to their conditions that they're in, and they're doing it from the poles to the equator in every single environment in the world. Remember, there's no time for natural selection to produce that variety, so the organisms are key. The organisms are the ones that are in control of everything they do. This is what we're doing too, okay? We do this with our bodies in every condition that we go into as we age and we go into different environments. The model we're using to test is called Astyanax Mexicanus. These fish come from Mexico, northeast Mexico. This is called the surface fish. So this lives out in the rivers of northeast Mexico, in the sunshine, where there's plenty of food, plenty of predators, seasons, different water, and so on. It has a visual system with a functional eye and distinct pigmentation. This is called the cave fish. This fish lives underground, miles underground, in total darkness, hardly any oxygen, hardly any food, and is completely thriving in the dark of a cave with a lot of other animals. These two are the same species. There's 30 different populations in Mexico of this and about 6 or 7 of the surface fish that are out there. And we're told that between 8 million and 200,000 years ago, this fish swam into the cave and became this fish. I'm going to show you that's wrong, okay? But this is currently considered the fastest evolving model in evolutionary biology, okay? But thank you. And so this is the cave fish, this is the surface fish. And we're working with these right now.
What's the scale?
Scale's about 3 inches. So we breed these in an aquarium. This is yarn, it's like a mop, and there's a grate on the bottom, and you're going to see a couple of fish come together. One of these fish is going to come around the corner. Oh, there you are, up there. I'll play this again for you. See the eggs coming down?
Wow.
Okay, let's do that again. So right here, right there, these two fish find each other. They're going to the top. Immediately they spawn the eggs. Sperm comes out. They're already fertilized as they're falling through the water. They're going to come down to the bottom of the tank. They're going to go through this grate. Some will be eaten along the way, but most of them will make it through, and this goes on for hours. So a couple hours, we can have 500 to 1,000 eggs in one spawning. You have to have reproduction or you can't have a system to be tested. So this is our model. What you get is all this developmental material. So my specialty is developmental biology, and I study how you build an organism. How you go from a fertilized egg and put all the right cells in the right place at the right time, and then express all the right genes in the right cells to make muscle and nerve and digestive systems and everything else. It's marvelous. It's like looking at the creation all over again. The most potent cell in the world is the egg cell. It's capable of making an organism from itself that is capable of making more organisms and more organisms and more organisms. And in every single organism that's in the multicellular animals and plants, the development goes through a series of stages. So what you see here is 2 cells, 4 cells, 8 cells, 32, close to 100, and all the way through. And here's a larva that's inside this case right here. And then in about 28 hours, it'll hatch out. And it lives, this is the yolk, and here's the head and the ear and the body. So these animals grow around a source of nutrition like we do. It's an extra embryonic source of nutrition. Birds do the same, mammals do the same, and fish do the same. And here's one of these guys at 7 hours after fertilization. We already know where the anterior is, the posterior, the left, the right, dorsal, and ventral. Okay? And all of this material, these are all cells coming down over the top to produce this animal. It goes from fertilization to a swimming larva in 24 to 28 hours. That's pretty fast, but most organisms that people study, like fruit flies and nematodes and urchins, they can have a whole generation in 2 weeks. These will take about 5 months to be able to reproduce. So it's a slower system, but it's much more complicated because it's a vertebrate. And here you got a beating heart, the tail's moving. So this is a video of these larvae that are blind, and they're developing, and they're about to hatch. So just to show you what we can see under the microscope, I can look in here and I can find the eye, I can see the pigment, I can see the ear, I can see what's going on early in development. Or I can stop them at different stages and study them more in depth. Okay, so eyed surface fish likely swam into caves and became the blind cavefish with distinct sets of optimized adaptations. So this is what we think of as the organism is in control of all the changes it makes. That's the process of adaptation. These fish have the capacity built into them. When did this happen? It happened after the global flood. Where they live is in caverns, in cave systems, okay? That's calcium carbonate. These are the caves all over the planet. Carlsbad Caverns, Oregon caves, all through Missouri, Kentucky, and so on. Why did they surface go into the caves? Because that's what God gave them in the original mandate. Be fruitful and multiply and fill the earth. So we have a major judgment upon mankind. God floods the planet. He creates all the sediments. And then as the waters wash off, they carve and sculpt and scour and change the whole shape of the earth. They also cut through this cave material and make big caverns and leave water behind. So this is approximately 4,370 plus or minus 5 years after the flood, or after creation. That's when this happened. So these are brand new fish going into a brand new environment. They're not the only organisms in that environment. Can surface fish and cavefish reproduce? Yes. Those 2 fish I showed you are the same species, which means they can reproduce with one another. And a small percentage will have eyes, most of them will not have eyes. But you have a good genome in one where it's not under regulation, and the genome in the cavefish, which can't make the eyes. And I'll explain that as we go. Then how could they survive in total darkness? This is the key to everything. This is the adaptive process, or adaptation. Compensatory adaptation. So with the transition from sunlit rivers to the darkness of caves, hypoxic, low oxygen, hardly any food, dark rivers, adaptive changes would have happened in all the major organ systems. Feeding would be enhanced, smell would be enhanced, respiration, energy, and sensation. I'm going to take you through some of these. So we predict that with a loss of vision, compensatory adaptations are predicted. Think about people. People that are born blind or become blind through disease, they have senses that are optimized, that are heightened. They can sense vibrations much better than we can. They have a sense of smell that's more acute than ours, right? They can hear better. All of their senses are compensating for the loss of vision. They have adapted with what God gave them. So these other systems now get more enhanced, more optimized, and they compensate for the loss of vision. That's what's going on in these fish. In fact, that's what's going on in every single organism on the planet if you start to look at where they live, how they live, and when they became what they became. So, starting with feeding, this is gustatory. The picture you see here is of a cave fish. It has no eye, and here's a surface fish. And these are taste buds that are now outside, all over the nose, all over the head, and outside the mouth. This is hundreds and hundreds more than the surface fish. They've got to taste, sense every little morsel of nutrition in that cave. Remember, in a cave where you're blind, you're at the mercy of a lot of different senses that have to help you, and there's hardly any food to begin with. So anything that will help you feed is really important. The first thing you have to do without an eye is you have to eat. If you and I stop putting nutrition or nutrients from outside our body inside our body, we're done. The experiments that the Irish did a century ago didn't help them. They lasted maybe 26 to 27 days, 30 days in their protests. But that just shows you humans can only go so long without eating. These fish are optimized. They've got more taste buds in the adult fish. They have extra oral taste buds on the outside. The jaws are bigger. There's more teeth in the jaws. There's more sharp points on all the teeth compared to the surface fish. Remember, this is compared to the other fish. This gene gets expressed and increases jaw size when you knock out the lens. So if you turn off the eye, by science, by experimentation, all of a sudden the whole feeding apparatus starts to kick in and change and optimize. So they're connected. This particular gene is linked to taste buds and jaw size, and this is the master regulator that knocks out the gene that builds the eye. So God has already programmed that when you go in these caves, I'm going to turn off your eye because it's 15% of all your energy just to keep that eye going. And I'm going to take away the pigment. I'm going to take away the pigment because you're not being hit by UV light from the sun, which kills you and gives you cancer. But you have to eat. You have to smell. You have to do all these other things. And that's what's going on there. In smelling, they have larger olfactory placos. This is highly innervated right here. So this is acetylated tubulin from a confocal laser scanning micrograph. And I'll show you that too, because this is one of our major resources. So you have nerves going to the upper jaw, nerves going to the lower jaw, all the way through the forebrain, midbrain, hindbrain, and down the back. The olfactory epithelium is larger. The nerve going to it is longer. They respond to much lower concentrations of nutrients like amino acids in food. Again, this gene right here increases neural populations in the olfaction, so it's compensating for not only the teeth and the jaws, but also smelling the food that you need to get to those teeth and jaws. And then the cave fish are far more efficient at finding food in the dark than they are the surface fish. Their blood, respiration, they've got erythrocytes are larger in the cave fish than the surface fish. Cave fish, unlike us, they have blood cells that have nuclei, so they can transcribe molecules, make genes. In each of the blood cells of a cave fish, it's larger, and they have more hemoglobin in each cell than the surface fish. So if you have a cave with very little oxygen, you need to pick up every single molecule that we're doing right now automatically by breathing. We're not even thinking about breathing. In fact, when we were born, the concentration of gases on the planet was perfect. You took your first breath and everything started going, and you probably haven't thought about it since, but you're 5 to 6 minutes away from death every 6 minutes, because if you're without oxygen, that's how fragile we are. There's 240 intervals of 6 minutes in a day. There's 37,600 intervals of 6 minutes in a year, and you can tell the gospel to someone in 6 minutes and save their life. But if they're without oxygen, they have a problem. So the life is in the blood, that's where it is. The hematocrit, the number and amount of blood with oxygen in it is much higher in the cave fish than the surface fish. So they're optimized. Hearts are larger. They transfer oxygen faster across their hearts. They've even changed shape. Higher oxygen. Larvae and adult cave fish have a slower heart rate. They don't school anymore. They don't sleep anymore. Okay? Major changes. Energy. This is amazing. These fish store high amounts of body fat. High triglycerides, way up there. Lipoproteins, way up there. Fatty acids, way up there. They're insulin resistant. They're hyperglycemic. They're all the things that our physician would tell us it's time to do a new exercise and a new diet. These fish are thriving on this. They can go for 6 months without eating, and they live as long as the surface fish. Okay, these are adaptations that allow these fish to thrive. By the way, there are 230 species of cavefish worldwide wherever we look for them in caves. This is not a one-off. This is a pattern on the earth. And then this is the one I'm going to talk about because I think this is one of the most important ones that I've been doing the work on, and you saw this from the very beginning. So the sensory system and all the sensory systems that are connected to it are critical for this fish to live and navigate. What you see here is a schematic of what are called neuromast cells. They're nerve cells, and the neuromast is sitting outside the skin. Okay, so you have little cilia up in the water, and they're inside a cup, and they have cells called hair cells, and it goes to the heart or to the brain and back out. And they're designed to detect the motion of water. So if a fish is swimming, it's pushing water by its body, and it bounces off the cave and comes back to it, and it senses that, and it goes left or it goes right. Because if it banged into the walls of the cave, it would scrape itself, it would get wounded, it would die. It can detect a little bug that falls in the water that's fluttering, because that's food. It can find its passage through an obstacle course in the cave. And it can find its mate during reproduction, and both of them are blind. How do you do that? How do you avoid a predator? So this would also have to happen really fast, a very fast adaptation. And that's what these are. If you look closely here, this is actually the eye of a cavefish going all the way around. And the yellow little balls and the cilia sitting up from them are these. And the muscle is in the red underneath them. And that's right here, but it's not shown. And then here's the nervous system that's connecting all these one to each other and back to the fish's brain. So what they do is they detect mechanical motion by the water. They send it into the nerves, and it becomes a chemical signal that goes to the brain as an electrical impulse, and the brain immediately says, turn left, turn right, swim up, swim down, go the other way, do what you have to do. Far more sophisticated than a nuclear submarine or anything that can fly in the air. Optimized, completely optimized. Think of it this way. These guys have hundreds of satellite dishes or antenna on their heads, picking up every single signal. They're navigating the entire underground water system with their nervous system. That's how they see. They're seeing with their nervous system. That's the way people who are blind do too. Okay? They navigate in their world with help from us, but also with their nervous system. If you look at the two fish, here's the larva of a surface fish with an eye, a surface fish, and then a cavefish and a cavefish. And all these are these hair cells that are all over the face. Now they've got hundreds of little satellite dishes and TV antennas on the outside of the head, just like the taste buds. This whole apparatus in the anterior is the one that's communicating to the fish where I am, where I'm going, who's in front of me, who's coming behind me, and so on. It's radical. And here's what they look like up close. So these are confocal laser scanning micrographs. So I can fix these fish at certain stages. I give them a cocktail of different chemicals, kind of get them drunk, and then I knock them over the head with a fixative, and so their morphology is perfect. We're talking about anything from 1 millimeter, a little bit less, to 2 millimeters. And then I will incubate them in certain molecules, fluorophores, that bind to muscle or DNA or nerves or whatever it is I want. And they will stay there, and then I can put them under this microscope, and it's like an MRI for little things, and it goes down through it, laser by laser, slice by slice, and it gives me all this information. So we're looking now sub-tissue, subcellular level. This is at 400 times magnification. Each one of these gray dots is the nucleus in an individual cell. Each one of these red boundaries here is the cell boundary, the cytoplasm, or the cytoskeleton that goes around the cell. These are the cilia, and the red is the muscle underneath. And here's what it looks like in the ear. This is very much like our ears, where you're listening to me right now, and these little guys are getting stimulating and sending something to your neural circuitry turning it into words so you can understand in real time as fast as I can talk. It's a good thing I don't talk real fast. We'd all be going like itching. And then we look at the fish, and here you see them. These are the neuromasts, all bound by nerves around the jaw, under the chin, a very highly ciliated nose, up in the ear. And then we put the other markers back on, and we can see the clusters of cells. We can see this big muscle that operates the jaw, opening and closing. We can see that the eye is now deteriorating. In fact, their lens starts to deteriorate between 12 and 24 hours of development. It's already programmed to turn off. And when it dies, the retina goes. And when the retina goes, the optic nerve goes. So God has already programmed these fish that you're not going to use an eye, but I have to put it in place to begin with because we have to have all the circulation to all the parts of the head. We have to put all the nervous system that connects everything, and then we can turn this eye off, and everything's in place to do all the other sophisticated organs and organ systems and tissues at the same time. And then we look down the top of the head. Here's the nose. Here's the brain. Here's muscles. Look at the symmetry. 1, 2, 3. 1, 2, 3. 1, 2, dip 3. 1, 2, dip 3. Perfect bilateral symmetry. This is looking up from the jaw, underneath. All these are in symmetry. So here's the nervous system. These are the neurons and the neuromass. And here's the muscles that operate the jaw. Here's the DNA and so on. And we look at the eye now. This is 9.5 days and 10.5 days. And this is where I'm focused most of my attention right now is looking at this blindness, how it happened, how we might be able to restore that. We do want to give sight to the blind at some point, but we have to do it through the mechanism of science and a lot of prayer, by the way. We need your help with that. So here's a deteriorating eye, and here's these big nerve cells. These are the neuromasts. They're going around the eye, but they're not in a perfect circle. Here's the surface fish that has an eye, has a big nerve. This is the retina, and all of these neuromasts are like the numbers on a clock, perfect symmetry in a circle. These are already designed to start moving into that area so that when the tissues grow over after the eye is gone, they'll be in place to fill up that head with more antenna. And this eye right now, at this point, the lens has already fallen out of the retina into the water. The retina is deteriorating, going through cell death, okay? And then it's going to start covering that tissue. And I want you to think about this. The lens itself is key to vertebrate development, especially ours. There was experiments done 32 years ago where they went into the surface fish, took out the lens, which is in the center, and put it in a cavefish at 24 hours, and it started to cause the nerves to grow back, and the retina started dividing. They took the cavefish lens and put it in the surface fish, and it caused cell death. So that lens is key to tell the rest of the organ systems, I'm going to build an eye here, and all of you are going to do the job that you're designed to do. The gene that's supposed to stop cell death has been downregulated in the cavefish, and it's regulated in the surface fish on purpose. Remember, as I go through this, none of the genes I talk about are missing or mutated or lost. They're all in the fish. The secular world has already found them. They're just under regulation. They've been moved to different locations. Okay? We look up closer. Now you can see how big these are. Okay? A lot more hair, a lot more cilia. And if we go look at 25 days, look at this. Look how many hair cells there are here and how big they are compared to these little guys. Look at the size of this nerve right here. It's got multiple cables in it, and here's the one in the surface fish. So this guy's using his eye, this guy's using his nervous system to compensate for all of that. And so we've done a study of eye development right from the beginning. This is the lens in a cavefish that's already down toward the bottom, not in the center, and it's going to die. And here's 5 days, it comes out. This is about a week and a half, the retina's tearing, 2 weeks, and then by 3 to 4 weeks, all this material is either falling into the water or falling back into the head, and the tissue will grow over. And here's another look. So this is an image of a 3 days development of cavefish. Here's the nose, the eye, the ear, the yolk, the heart. There are 11 different images that have been put back together in a stack, so it's all in focus for you. And here's what the confocal microscope does. So we're now going through a scan. All the white dots is DNA. All of the red are cytoskeleton and muscle filamentous actin proteins that are between organ systems and laying out the different parts of the nose, the forebrain, the midbrain, the hindbrain, and so on. You see these little, here's your neuromass coming up on the outside. And then all the yellow is the nervous system. And I can stop it and take the colors off, and then I can look up close and you can see all the pattern. So in other words, this is modular. You can turn off a system. I can go back to this plug back here and put in a refrigerator, a TV, whatever you want, as long as it's got power to it. Plumbing and electrical, same thing. I can take off the eye as long as I've got power to the other things, but you have to put it in place to begin with. So you could pull an organ system out and you can turn it back on. And then here's at 400 times magnification just looking at the eye. Here's where the lens is. This is the retina. Here's the optic nerve. I'll let that come through again. This is really cool stuff. We have the capacity to look at this. So we're looking at from macroscopic all the way to microscopic and then at molecular level, which I'll tell you about too. And here's this lens forming. If we look up close, all these little white dots, that's DNA coming out of the cells that are dying. So I know right away this is a cavefish, and I know when it's starting to decay. So we have an understanding of the developmental timing of when that eye gets turned off and the retina gets turned off and so on. These fish survive. They're thriving. You saw all the other adaptations. So this is not about a dying fish. This is about reconstructing the priorities of your nervous system and your digestive system and everything else to compensate for loss of vision. If we look, this is the muscle, the same type of fish, cavefish at 3 days. And here, I've used a histone marker. So these are antibodies that go in and they bind to part of the DNA or a molecule that the DNA is wrapped around called histones. And they only do it to the cells that are undergoing cell division, which tells you it's a live cell. Cells that don't longer divide are either done or they're dying. What you don't see is this going on in the lens. So this lens is dead and dying. Here's two sets of chromosomes dividing. There's another 2 sets of chromosomes dividing, and so on. So all this tissue out here is alive. The retina is still alive, but it's going to be going away. And then the lens itself is not alive. And if you look at them close, back and forth, here's the little markers that show that this is DNA that's coming out in little vesicles. And then here you can see it matches up with the lack of green. In the surface fish, here's the lens area, and there are live cells inside of it. In fact, there's live cells all the way through the retina, and all the nerves that are supposed to be there are still there. This is a functional surface fish to compare. So now I can look at, you have to have perspective. One's green, one's red, one's tall, one's short. Once you have that perspective, you know how to compare one thing to another. Eye formation is integrated with development in all major organ systems. That's the first takeaway. The secular people say that the apoptosis, the loss of the eye, starts at 24 hours. And then by 35 hours, the retina starts to do it. I think this actually happens within the first 12 to 24 hours, probably earlier than we think. Most things actually happen before we see them, by the time scientists come along. The expansion of this particular thing, this molecule, sonic hedgehog, as I told you, results in cell death, lens death, and arrested growth. Epigenetic silencing, I'm going to tell you that in a minute, but most of these genes that build the eye are under methylation. It's a marker that's on the DNA, and it closes up that area so that none of the machinery that goes in and turns the gene on can get there. It can't start transcribing all the things that build an eye. That's another pattern of God regulating. In fact, think of God's government. I want you to think of this for a minute. powers, principalities, the angelic beings. He has a government. Everything else in the universe is chemistry. They're atoms. His government reigns. On Earth, there's governments. Our government in the United States is divided right down the middle right now. But it is a government. There's county governments. There's governments in little cities. There's even a family unit which has a governing body, the parents over the kids. That doesn't always work out, but that's what God put in, the family unit. In the molecules, in the cells, there are governments. It's called cell regulatory networks, or gene regulatory networks. And there's genes that are master regulators, and they control genes below them and tell them what to do. And then you have other genes on the periphery that go and build that fin and build that arm and build that wing and so on. But there's genes that are really powerful in different levels. And then the lens transplantation, I talked about that, demonstrated potential. The secular world believes that they could restore the cavefish eye, but they haven't done any of those experiments. In fact, the first experiment we did when we got these fish is we put them in the light, we put them in sunshine and artificial light, and we put them in darkness. The world, the evolutionary biologists, only put them in the dark. There's a message there. And all of this is backed up by their work, everything I'm seeing here, okay? We're seeing this, but I just want to show you, I'm not making any of this up. This is what they're finding. Okay, we're headed now to a brand new area in this work. And here's the message. It's the tiniest little things in the world that manage and regulate the biggest things in the world. Wow. Inside you, molecules, biochemistry, cells, in each one of your cells is controlling what's going on. It built you, it sustains you, and it's holding you through until sickness overcomes you or whatever happens. In each one of us, there's about 20 to 30 trillion cells. That's a lot of cells, and they're all regulated. Omics is the characterization of everything that's has to do with molecules in living organisms. Proteomics, genomics, transcriptomics, and so on. So we're going to start doing this. In fact, before coming here this week, for 4 days, every 3 to 4 hours, I collected the cells and the embryos and the larvae at every stage in the first day, the second day, and the third day, and put them into a storage freezer, -80, super cold, in a reagent that preserves them. And in about 2 weeks, we're going to send that off to a big company, and they're going to sequence all the DNA and all the RNA and everything that's in those. And we're going to get a map of what's going on in these fish at the molecular level. This is cutting edge. This is what all biologists do if they're actually looking at a model organism. So you guys have heard this first. This just happened this week. I didn't get very much sleep because I was up till 11 and back up at 4 to get the next setup. But we had a table like this with dishes and dishes and dishes of fish at all different stages. And we would collect them and preserve them. And we're going to do the same with the surface fish too. So this is the next fruitful area. And this has also never been done in the history of ICR. In fact, what I've shown you is all brand new to ICR. Molecular biology never had a lab. Microscopy, imaging center never had a lab. Raising animals never had a lab. No other group in creation science is doing this. There's people at different universities that are taking part in that kind of work, but we are actually leading this. You heard from Tim Clary, I think last month, and he talked about geology. That whole characterization of the geologic column worldwide is the most comprehensive geological characterization that anyone knows of. So we're going to sequence the genomes, which is the structure, function, and mapping of every gene. We're going to look for the markers that are regulating, like the methylation marker I just told you. And then we're going to look for all the transcripts. So transcripts is DNA codes for an mRNA. DNA is a double helix. It opens up, and one side of it gets copied, and it gets sent off into a ribosome, and it gets translated into a protein. That's the basic molecular biology. Today, the RNA world far surpasses that. We can think of a single cell going through enough activity in one day that is what it takes a major city in America to do in a year. shipping, receiving, packaging, communications, quality control, recycling. They're doing it like this all day long. That's one of the most complex units in all of biology. In fact, the cell is the basic unit of life. And until you have a cell, you don't have life. The candidate genes, we've got transcription factors, growth factors. These are all involved in the forebrain and the optic cup in the retina. Eye field specification, anti-apoptosis, we want to turn that lens back on. So we're going to be looking at the expression patterns of all these genes, learning from the secular world in all different animals as eye development has been studied. And here's just a table of all these papers and all the different genes that the secular world has been using, because they make these available in our papers. And remember, our scientists have walked in the shoes of the secular team, right? We've been to the major universities. We've done a lot of research. I've probably done the most extensive research. I was with the Smithsonian Institution for 9 years. That was kind of my reconnaissance. I was gathering information until I came to where I am. But we know how to read the methods and materials. We know how to use the instruments. We've got access to all the work that the world is doing. We just have a very different worldview. And we give all the credit to our creator. So that's the main difference right there. So this is sonic hedgehog I mentioned. This one's responsible for regression of the eye. This one is called pair box 6 or PAC6. Now think about this. This gene has been studied for over 40 years. It's the master regulator of eye development in every animal where it's been tested. This gene has such power that I think it's probably 35, 36 years ago, they took the sequence that codes for the PAC6 gene out of a mouse and they glued it into the same place inside of a fly, in the genome of a fly. And then they took that and injected it into the antenna and the leg and the wing. And wherever they injected it, it built an eye. This gene is such a master regulator, it goes into that situation and says, stop what you're doing, start making all the things that you need to build an eye system here. So that's one of our candidates that we're going to start to look at. Maybe we express it at the wrong time. Maybe we stop its expression and see what happens. Maybe we turn it back on in the lens at 2 cells or 4 cells and it overrides the signal to stop it, and the lens starts to develop and the retina starts to develop and maybe the eye can be restored in the cavefish. That doesn't mean that's the natural way to do it, but it shows you where on the power grid the big fuse boxes, the big levers are. That one with the red handle, we want to pull that red handle and find out what's going on. So that's really important. This one's involved in lens development. Paired-like homeobox, tooth and eye, and all these things are real important. And then last year in June, these folks, Chenard et al., used CRISPR, the gene editing program, and they went in and took this RX3, retinal homeobox, and they knocked that gene out in the genome of a surface fish, and the surface fish developed without an eye. So there's a major candidate right there. If this one's not being expressed, that might be the master of the master. Who knows? But that's the idea. That's how this whole study kind of works. And then this is the whole RNA world is getting huge, and I know you're not going to be able to follow this, but there are approximately 14,000 more of these in the cavefish than the surface fish. So there's all kinds of other molecules that are being turned on in these fish to compensate and help build that body plan and keep it alive compared to the surface fish. And then this is the slide that does it all. Eel, salamander, leech, bugs, millipedes, centipedes, spiders, crayfish, ants. All of these are found in caves. They're all blind, and they have little pigment or no pigment. Folks, this is divine engineering. God programmed all these animals, when they're in that environment, shut down the eye, shut down the pigment, thrive. They're reproducing in the caves. They're living in the caves. They're living in caves worldwide. Be fruitful, multiply, fill the earth. I need my biology in the cave system too, so go do what I do. Humans don't obey the mandate, but the animals do exactly what God tells them to do. And that's what's going on. So, cave-dwelling adaptations are observed in all these different animals. There are 230 cavefish as of 2018 around the world, so it's not a one-off. This cavefish is has essential optimizations to compensate for the eye loss by enhancing all of these systems, and they're all promoted as convergent evolution. Convergence means that two completely different, unrelated lines of animal development came across through life and came to the same adaptations. But the surface fish and the cavefish are one species. It can't be convergent evolution. These might be convergent with the other animals in the cave, but they use these words. In fact, here's an unknown secret to most of you, is that the jargon, the terminology within evolutionary biology and biology itself is all kind of fabricated to make the story fit. They ignore a lot of things, but they have natural selection. In fact, natural selection has 7 or 8 different flavors. There's positive selection, negative selection, neutral selection, directional selection, fluctuating selection. Whatever they need to explain what they're working on, to validate their case, they have the words for it. And I looked at this for years. In fact, I didn't even go to college till I was 36 years old. Okay? Wow. I went in there with a prayer. God, take me backstage. Show me how they think. Show me the whole theory of evolution. I need to study it right alongside them. Let me use the technology that they have. What are we looking at underneath the microscope today, Father? Right? For years, I was almost 28 years behind enemy lines, you might say. They never knew what I believed because they would discriminate, right? It's amazing. People discriminate by what you look like, the color of your skin, your ethnicity. But as soon as you open your mouth and you tell them you have a different worldview, that's the worst one. They're discriminating about what you think or what you believe. That's the war we're in right now. That's why it's important for me to come and tell you about this. We're doing the same work. We're looking at it a different way. We have a very different explanation, and the implications of that are profound. Okay? Only one creative power can account for this. Every molecular, cellular, organ system, component of life, and he is before all things, and by him all things consist. Thank you for your time.
Oh.
That was wonderful.
Thank you, Doctor.
Questions?
Yeah, we're going to open the floor to questions, so if you would raise your hand, and I'll call upon you so we don't step on each other. So, and then Dr. Boyle will be glad to answer. So, yes, sir.
So I understand correctly that the surface fish was an active 6-day creation, and the cavefish evolved, adapted from that.
Adapted from the, yes.
Do you have any idea how long that process took?
They tell us that it took between 8 million and 200,000 years for the surface fish to go in and go through natural selection, mutation by mutation, accident by accident, until it cobbled together and built that fish. They would have died and died and died until one of them somehow survived its injuries or found enough food to do that. I believe this would take at most 1 or 2 generations. Because all the environmental parameters that the fish are tracking with continuous environmental tracking, they're going to assess all those conditions, and then internally, the body's already regulating, get ready. In fact, you see this fish right here. This is a cave fish that was born in the light, under high intensity light, grow lamps, and after 2 months, it took on all the pigmentation of the surface fish. So the parents...
The cavefish went to a surface fish?
It doesn't have an eye, but it got all this pigment. Yeah. So in the light, the parents were already exposed to the light. And they're probably passing on through epigenetic regulation. I want my kids to be ready for, when they get out, they're ready for the world they're exposed to now, because they're no longer in a cave.
Right.
In one generation?
In one generation. So I think...
Well, you've duplicated this.
Yeah. Well, we haven't duplicated the eyes. So we've raised 3 generations of cavefish in the light, and they haven't made eyes. They've gotten tanner, but now this is not really important because all fish can do this. Wow. In the nighttime, in the daytime, if they're stressed, they will change their colors. They will do things. That's not a big deal. We thought it was something important because it was interesting, but that really doesn't change it. But these fish are already programmed to make all the changes. So I believe if I was to take a bunch of cave fish and go down to Mexico, outside the cave in the river, and build a pen that they couldn't get out of, where they have all natural sunlight, all the photosynthetic different molecules, all the different foods, all the perfect nutrition, the seasons, everything else, and they're away from the lights and all the magnetism that the city's doing and everything else, they would probably become surface fish within 1 or 2 generations. Might even the first generation. Because it's all everything that's there works. And here's why. These folks, they rely upon the fact that the cave fish and the surface fish are the same species. And they do hybridization with them. And a certain percentage comes out with eyes, and a certain percentage doesn't. That means that the cave fish, the babies, still have all the biochemistry that's ready to go, and it works. All you need is one set of chromosomes where it's not under regulation, and the rest we have, and now we can make fish with eyes. So if they could do that by hybridization, all different caves, then you could probably turn the eyes back on. But we are not able to do this in the laboratory. We've tried changing temperature, pH, salt content, you name it.
How long is a generation?
It's we've ramped it up to about 5 months by feeding them high protein 3 times a week, but naturally it's about 10 to 11 months. So you have to wait a long time. But we can do all these things to them and spawn them and see what happens. Do things to them and spawn them and see what happens. But that's not working. I do have a box of cave rocks, finally, and I'm going to try to mimic, because I think the calcium or the chemistry in those caves is really key to all of this. The fish that are in the water, in that cave, and all the chemistry coming off the walls is probably triggering this change. The animals that are on the walls have the same output. They're blind and they have no pigment, but they're still reproducing. In fact, outside the caves, all those animals have eyes and all the different colors. So something's happening inside that cave, and it's not just darkness. We've kept them in the dark for months, nothing changes.
So you're grabbing baby fish from the cave.
No, we're not. We're now only getting the fish we have been the cave fish have been raised in America for years. We get them out of labs. You can get them from Amazon if you want to do something. They'll come right to your house. They'll deliver right to your house.
You can get anything from Amazon.
But we recently got a surface fish that came from Mexico only 1 generation ago. It's brand new, and we have access to that too.
So it's cell lines that you
Yeah.
So you have a question, ma'am?
Yes, I have a comment and a question. Do you sell any of your literature? Because my phone was dead. I left it down here. I left it with a friend. So I had no way of taking pictures. So do you have a book that I could buy that has
your information.
your information in it?
The information on the study I just presented?
Yes.
I don't have a book out yet, but I do have a copy of Acts and Facts with a special issue on all these adaptations.
So can I?
I have one for you that's free.
Okay, I do have.
Which leads me to one other thing. This is the book. This is the magazine. These are free. If you go over to Benjamin and sign up, we'll give you every other month. You'll get Acts and Facts. And you didn't get it?
No, I got something right here.
Okay, there it is.
What's that?
Yeah. And so we have a lot of different resources. Henry Morris wrote 66 different books. There's all kinds of stuff that's coming out new on cell biology and so on, but we have not written a book on this yet. However, our director, Randy Galuza, the president of ICR, was an engineer. He was an MD. He was 21 years in the military, surgeon, and he is the one who's promoting the engineering, but he needed a biologist, and so he found me. Actually, we found each other at a conference one day, and that's how I got to ICR.
Well, I feel like since I'm a pastor's wife, or was one, my husband was an ordained minister, and you know, it has all kinds of things in the Bible about creation, and my husband studied all the time. He could tell within a few scriptures where it was found and all that. But all my kids are really trying hard to live for the Lord, and this is one thing, my son is a worship leader at Happy Trails Calvary Church, and it's weird how God's made it where other senses will heighten if they're blind or deaf or whatever. Because I've worked in a nursing home as an activity director, so I know even as Alzheimer's, they still have quality of life.
They do.
They do, but like the fish, the cavefish, I tell you, my sons raise blind dogs, and they can do anything that the others can do except for say, "Oh, woo, woo, oh, woo, woo." They bark. All of the other dogs bark, but that little blind dog, she can skateboard across the floor just to get a treat.
Yeah.
She can stand, as heavy as she is, she can stand up on her hind legs. So I'm just saying, even the guy that sings with my son, that's why I mentioned him, he's totally blind. He knows exactly how many steps to get to up to the pulpit, and my son helps him back down, but because he might sit on someone on the first pew. But I'm telling you, it seems like if you're deaf, if you're sight, or
Right.
whatever,
If we
it
turned
heightens.
off some of the noise that's coming into our minds in our lives, we would have time to concentrate on these things. We'd know a lot more. We'd be more efficient in everything we do every day.
Right.
And Lord is trying to tell us, stop listening to the world. Concentrate on what I'm telling you to do. So it's the same principle. By the way, I'm promoting this book especially. This is an amazing book. This one is talks about the long war, from the sciences, from the schools, from all kinds of different disciplines within society. And this one by Henry Morris, I've got a whole big shelf full of commentaries. I've got 14 to 15 different commentaries on Genesis alone. This is the best commentary, not only on Genesis, but on everything that I've ever seen in one Bible. His commentary on Genesis and the creation, all the way through the scriptures, from Genesis to Revelation, is by far the most, it's amazing. It's not commentary by commenters talking about what other commenters said, which is most of what your commentaries do. Who said what and what they think and so on. Henry just tells you it's electromagnetic radiation, that when God said, let there be light, there's light, and here's why, and here's entropy, and here the plant. I mean, think about this, the plants. The plants were created on day 3. Plants photosynthesized. The sun wasn't even up yet. You can't go for days without the sun. So the next night, or that night, they're already doing oxidative phosphorylation like what plants do at night. It's a special time for plants. They do it every single night. The sun comes up in the morning, the leaves open up, the photosynthetic antennae are ready for the sun, and they start to... So there can't be any time other than 24 hours right there. And 2 days from that point, here comes all the flocks and herds on the planet, and they're going to have to eat. And they're all eating plants and vegetation at the time. So we're talking 24-hour days. And he says it with ordinals. He says it was evening and morning. Evening and morning the first day. Evening and morning the second day. And as far as the stars and molecules, I'm almost done.
You're fine. No,
And then
I'm listening.
he made the earth on the third day, separated from water, and the plants are there, and so all the atomic nuclei, every single element in the world is there in soil and in the plants and everything else, and he makes the stars on the 4th day. So the stars are not the nursery for life. They're not the atom factories that the world tells us about. God does that too, and it's subtle. It's just you might not see it the first time. But he's saying they're going to ask this question, and I'm going to show them that it can't be that way. So he puts it the next day, and on throughout the scriptures.
Do you have any other questions? Yes, ma'am.
Just in light of the explosion in the past recent years, and this year, of AI,
Yeah.
how is that impacting your research and things, or how's it being incorporated? Is it valuable, or, you know, I mean, is it even, you know, what's it going to do in the research arena?
So right now it bothers me because every time I pull up Google and ask for something, I can't get rid of that page.
Because it's forcing us.
So you don't have to look at it.
Right.
You don't have to look at it. But I'm starting to look at what the terms they are using, and then I follow those terms and I go to the primary literature to make sure it's real. So it's doing things faster, but the whole idea of this is profound. It's prophetic, in fact. It is going to be where everything will be electronic and there'll be automated information not coming from humans. And right now the world's going, this is faster and more powerful, and we can do so much more work. Well, they're going to end up giving their whole lives under the power of AI, which is artificial. And so that leads artificial, right? It's in the word, is deception. Matthew 24. What is the sign of the end of the age? Be careful that no one deceive you. And he goes, deceive you, deceive you, deceive you. That's what this is bringing in. We want to automate our lives. I mean, look at the technology right here. This is important, but it's running on man's code that makes it more efficient to do that. But I personally don't want to jump into artificial intelligence. In fact, right now, you go to Home Foods, I don't know if or Whole Foods, and right by the place where you put your credit card in, there's one where you can put your palm on it, right? And you do it once. You put your card in, you put your palm on it. From that point on, you go to the store and you just put your palm on it.
But God has not given us a spirit of fear, but of love and a
Right.
sound mind. We can fear all these things that are coming up against us, but God says not to have a spirit of fear. And there's a lot of stuff going on in this world.
It's easy.
So we better be ready.
Yes, especially the young ones. Because all the kids are all walking around like this with their phones, right? They're not talking to each other anymore like we did.
John Lennox has a good book on AI. It's very
I believe it. Yeah, he's a brilliant man.
Any other questions?
All right, thank
All
you so
right.
much for your time.
Thank you. Let's show our appreciation.